Chapter 7, Section 2, Eukaryotic Cell Structure. At first glance, a factory is a puzzling place. A bewildering variety of machines, buzz and clatter, people move quickly in different directions, and the sheer diversity of so much activity can be confusing. However, if you take your time and watch carefully, before long you will begin to identify patterns. What might at first have seemed like chaos suddenly begins to make sense. Comparing the cell to a factory. In some respects, the eukaryotic cell is like a factory. The first time you look at a microscope image of a cell, such as the one in Figure 7-5, the cell seems impossibly complex. Look closely at a eukaryotic cell, however, and patterns begin to emerge. To see those patterns more clearly, we'll look at some structures that are common in eukaryotic cells, shown in Figure 7-6. Because many of these structures act as if they are specialized organs, these structures are known as organelles, literally little organs. Cell biologists divide the eukaryotic cell into two major parts, the nucleus and the cytoplasm. The cytoplasm is the portion of the cell outside the nucleus. As you will see, the nucleus and the cytoplasm work together in the business of life. Nucleus. In the same way that the main office controls a large factory, the nucleus is the control center of the cell. The nucleus contains nearly all the cell's DNA and with it the coded instructions for making proteins and other important molecules. The structure of the nucleus is shown in Figure 7-7. The nucleus is surrounded by a nuclear envelope composed of two membranes. The nuclear envelope is dotted with thousands of nuclear pores, which allow material to move into and out of the nucleus. Like messages, instructions, and blueprints moving in and out of a main office, a steady stream of proteins, RNA, and other molecules move through the nuclear pores to and from the rest of the cell. The granular material you can see in the nucleus is called chromatin. Chromatin consists of DNA bound to protein. Most of the time, chromatin is spread throughout the nucleus. When a cell divides, however, chromatin condenses to form chromosomes. These distinct thread-like structures contain the genetic information that is passed from one generation of cells to the next. You will learn more about chromosomes in later chapters. Most nuclei also contain a small, dense region known as the nucleolus. The nucleolus is where assembly of ribosomes begin. Ribosomes. One of the most important jobs carried out in the cellular factory is making proteins. Proteins are assembled on ribosomes. Ribosomes are small particles of RNA and proteins found throughout the cytoplasm. They produce proteins by following coded instructions that come from the nucleus. Each ribosome, in its own way, is like a small machine in a factory, churning out proteins on orders that come from its boss, the cell's nucleus. Cells that are active in protein synthesis are often packed with ribosomes. Endoplasmic Reticulum. Eukaryotic cells also contain an internal membrane system known as the endoplasmic reticulum. The endoplasmic reticulum is the site where lipid components of the cell membrane are assembled, along with proteins and other materials that are exported from the cell. The portions of the ER involved in the synthesis of proteins is called the rough endoplasmic reticulum, or the rough ER. It is given this name because of the ribosomes found on its surface. Newly made proteins leave these ribosomes and are inserted into the rough ER, where they are chemically modified. Proteins that are released or exported from the cell are synthesized on the rough ER, as are many membrane proteins. Rough ER is abundant in cells that produce large amounts of proteins for export. Other cellular proteins are made on free ribosomes, which are not attached to membranes. Other portions of the ER is known as the smooth endoplasmic reticulum. Because ribosomes are not found on its surface, in many cells, the smooth ER contains collections of enzymes that perform specialized tasks, including the synthesis of membrane lipids and the detoxification of drugs. Liver cells, which play a key role in the detoxifying drugs, often contain large amounts of smooth ER. Golgi Apparatus. Proteins produced in the rough ER move next into an organelle called the Golgi Apparatus. Discovered by the Italian scientist Camillo Golgi, as you can see in figure 7-9, Golgi appears as a stack of closely opposed membranes. The function of the Golgi apparatus is to modify, sort, and package proteins and other materials from the endoplasmic reticulum for storage in the cell or secretion outside the cell. The Golgi apparatus is somewhat like a customization shop, where the finishing touches are put on proteins before they are ready to leave the factory. From the Golgi apparatus, proteins are then shipped to their final destination throughout the cell or outside the cell. Lysosomes. Even the neatest, cleanest factories need a cleanup crew, and that's what lysosomes are. Lysosomes are small organelles filled with enzymes. One function of lysosomes is the digestion or breakdown of lipids, carbohydrates, and proteins into small molecules that can be used by the rest of the cell. Lysosomes are also involved in breaking down organelles that have outlived their usefulness. Lysosomes perform the vital functions of removing junk that might otherwise accumulate and clutter up the cell. A number of serious human diseases, including Tay-Sachs disease, can be traced to lysosomes that fail to function properly. Vacuoles. Every factory needs a place to store things, and cells contain places for storage as well. Some kinds of cells contain sac-like structures called vacuoles that store materials such as water, salts, proteins, and carbohydrates. In many plants cells, there is a single large central vacuole filled with liquid. The pressure of the central vacuole in these cells make it possible for plants to support heavy structures such as leaves and flowers. Vacuoles are also found in some unicellular organisms and in some animals. The paramecium in figure 7-10 contains a vacuole called a contractile vacuole. By contracting rhythmically, these specialized vacuoles pump excess water out of the cell. The control of water contents within the cell is just one example of an important process known as homeostasis. Homeostasis is the maintenance of a controlled internal environment. Mitochondria and chloroplasts. All living things require a source of energy. Factories are hooked up to the local power company, But what about cells? Most cells get energy in one of two ways: from food molecules or from the sun. Mitochondria. Nearly all eukaryotic cells, including plants, contain mitochondria. Mitochondria are organelles that convert the chemical energy stored in food into compounds that are more convenient for the cell to use. Mitochondria are enclosed in two membranes, an outer membrane and an inner membrane. The inner membrane is folded up inside the organelle. One of the most interesting aspects of mitochondria is the way in which they are inherited. In humans, all or nearly all of our mitochondria come from the cytoplasm of the ovum or egg cell. This means that when your relatives are discussing which side of the family should take credit for your best characteristics, you can tell them that you got your mitochondria from your mom. Chloroplasts. Plants and some other organisms contain chloroplasts. Chloroplasts are organelles that capture the energy from sunlight and convert it into chemical energy in a process called photosynthesis. Chloroplasts are the biological equivalents of solar-powered plants. Like mitochondria, chloroplasts are surrounded by two membranes. Inside the organelle are large stacks of other membranes, which contain the green pigment chlorophyll. Interestingly, chloroplasts and mitochondria contain their own genetic information in the form of small DNA molecules. This has led to the idea that they may have descended from independent microorganisms. This idea, called the endosymbiotic theory, will be discussed in Chapter 17. Cytoskeleton. A supporting structure and a transportation system complete our picture of the cell as a factory. As you know, a factory building is supported by steel or cement, beams, and by columns that support its walls and roof. Eukaryotic cells are given their shape and internal organization by a supporting structure known as the cytoskeleton. The cytoskeleton is a network of protein filaments that help the cell to maintain its shape. The cytoskeleton is also involved in movement. Microfilaments and microtubules are two of the principal protein filaments that make up the cytoskeleton. Microfilaments. Microfilaments are thread-like structures made of a protein called actin. They form extensive networks in some cells and produce a tough, flexible framework that supports the cell. Microfilaments also help cells move. Microfilament assembly and disassembly is responsible for the cytoplasmic movements that allow cells, such as amoebas, to crawl along surfaces. Microtubules. Microtubules are hollow structures made up of proteins known as tubulins. In many cells, they play critical roles in maintaining cell shape. Microtubules are also important in cell division, where they form a structure known as the mitotic spindle, which helps to separate chromosomes. In animal cells, structures known as centrioles are also formed from tubulin. Centrioles are located near the nucleus and help to organize cell division. Centrioles are not found in plant cells. Microtubules also help to build projections from the cell surface, which are known as cilia and flagella, that enable cells to swim rapidly through liquid. As you can see in Figure 7-11, the microtubules are arranged in a 9+2 pattern. Small cross bridges between the microtubules in these organelles use chemical energy to generate force, allowing cells to produce controlled movement using the cytoskeleton.